Thank you so much. So again, my name is Laina Lusk. I'm a genetic counselor at CHOP, the Children's Hospital of Philadelphia. And I'm going to be giving some sort of genetics basics, a bit of an overview, but then focusing a little bit on UBE3A and why it's important in Dup15. So just quickly, no relevant disclosures to close, no relevant conflicts of interest to disclose, but I have done some paid educational lectures for genetic counseling programs in Ambry Genetics, and I'm on the scientific advisory board for the UGTH Foundation. So going back to some genetics basics back from biology class, our bodies are made up of cells, and in these cells are things called chromosomes, which are packages of DNA. DNA itself is a long string of letters, A, C, T, or G, and there are sections of DNA that code for particular proteins, which help compose our bodies and make up a lot of the functions in our bodies. So that particular section of DNA that makes a protein is called a gene. You can think of this sort of like a chromosome being like a recipe book, a gene being like an individual recipe, and then that protein being the thing you're ultimately trying to make from that recipe. So just going to use this analogy and some other things in the future. So focusing a little bit more on these chromosomes, these packages of DNA. If we were to look at a cell under a microscope, we would see all of these chromosomes laid out. This has some staining on it to make this little band pattern. But if we straighten them up and line them up together, you would have 23 pairs of chromosomes. You get one set from mom and one set from dad, and they're numbered 1 through 22. And then you have X and Y, 2 X's for females and an X and a Y for males. If we zoom in on one of these chromosomes, we can see a little bit more of the architecture here. In the middle, we have something called the centromere. In the top, we have the short arm, which is called the P arm. The P comes from the French word for short, which is petit. And then we have the longer arm, which is the Q arm. And Q also, it has a French word Q for tail, but also it's the letter that comes after P. So I've heard that's the reason why it's called the Q arm, just comes after P. So if we were to take even a deeper dive looking at this chromosome and trying to understand why do we always talk about all of these letters and numbers when we talk about Dup15, we'll kind of see that this is giving us like a mailing address of where in the chromosome we might be finding a change. So we might start with the chromosome number. So that's sort of like the country. So in this case, we're talking about chromosome number 15. We might zoom in a little bit more and look at a state, which could be that arm, so the P or the Q arm. We can zoom in a little bit more and start to see some of these different sections. So we might break up that into different cities. So we can break up the Q arm here into 1 and 2, and these are called bands. And I'll note that over time we've gotten better at zooming in, sort of like Google Earth slowly zooming in more and more to a fine-grained level. So these are each successive then subbands after this. So we might look at like a neighborhood and we see all of these subbands here. And then we can go even further and say a street address, and then even a little bit further on some sort of like an apartment number. So when we put these all together, that's where we get all these complicated numbers from. In our case, we're interested in 15q11.2 to 15q13.3. Again, each of those numbers is sort of separate because it's like bands and subbands. So that's how you can sound like an expert geneticist when you're talking to your doctors, is you say each of the numbers after the chromosome 15 together, separately. So Dup15q syndrome is specifically caused by an extra copy of part of chromosome 15 in this region we were just talking about. You can have a duplication where you have 1 extra copy for a total of 3, or you can have a triplication where you have 2 extra copies for a total of 4. This can be within a chromosome, which is called interstitial. It's kind of right next to the other one. Or it can be what we might call isodicentric, where there's an extra chromosome where these extra pieces are contained. Now, some lab reports might say isodicentric, we shorten that as IDIC. You might see other things that say a marker chromosome or supernumerary chromosome or a pseudodicentric or dicentric chromosome. They're all just different technical definitions of this, but for short, I'm just going to call it isodicentric or IDIC because we don't need to get too technical on what specifically it is. But in all of them, it's an extra chromosome. It usually contains 2 extra copies there. So as you can see, it's usually 4 total. So zooming back in on our specific region here, so we can see those sort of bands spread out. And within it, we have a lot of different genes. And again, those genes are those recipes that make the different components in the body. So the genes are really what's important in this area. And another thing to note that's in this area are there's these things called breakpoints. And these are places where the DNA can start a deletion or start a duplication or start and end of that deletion or duplication. So the duplication in Dup15 is mediated by these breakpoints. And this middle part between breakpoint 2 and 3 is what we would call the critical region or the core region or the Prader-Willi-Angelman critical region. It's this section that when you have an extra copy, you have Dup15q syndrome. You can have bigger or smaller, but it has to have this section to be Dup15q syndrome. So again, this is the minimum amount that you could have and have Dup15. It can go to breakpoint 4 or breakpoint 5. It could start earlier at breakpoint 1 and go to any of those other points. It can even start before that breakpoint 1. But the main thing is as long as that breakpoint 2 to 3 area is included in there, it's Dup15q syndrome. That is the most important region. Sometimes if it's bigger, it can be more severe, but it doesn't change what type of syndrome it is. It's still Dup15q syndrome. Another complicating factor when we're thinking about the genetics of Dup15 is something called imprinting. And that's where certain genes are only turned on if they come from an egg from the mom or if they come from a sperm from the dad. And they're not turned on if they come from, they're differentially turned on. So just showing a little diagram about this, most of our genes are not imprinted. So most of them, it doesn't matter if it's coming from an egg or a sperm, mom or dad, the genes are all turned on. This is kind of showing a purple gene and a yellow gene, they're all turned on. When something is imprinted, they get turned off when they're coming from either an egg or a sperm. So in this case, the yellow gene is turned off when it comes from dad, and the purple gene is turned off when it comes from mom. And that off switch is something called methylation. So in this case, only one copy of each gene is actually turned on and used in the body. The other one is turned off. So what happens in the egg and sperm, this pattern gets sent, they combine together, and then the child just has one copy of each. So in Dup15q, classically, it's caused by an extra maternal copy of this section. So that means the genes we're most interested in here are the ones that are turned on from the maternal copy. We do know that you can have symptoms if you have a copy coming from dad, but often it's a little less severe. So these are the genes here in green that are expressed from both chromosomes. Doesn't matter if it's from mom or from dad, they're all turned on. These ones in pink are the ones that are only turned on when it comes from the maternal copy. And then the ones in blue are the ones that are only turned on when they come from dad, or they're more highly expressed when they come from dad. Those ones are a little bit of less interest because we know it's this maternal copy that seems to be particularly important when it involves Dup15q syndrome, or it leads to a more severe phenotype. So thinking a little bit more about these genes and what genes should we target when we think about therapies. I just want to, again, kind of go back to our analogy a little bit. So we think of this chromosome as a book of recipes, and you can split them up. There's all these different genes in there or different recipes. So you might have a recipe for your favorite chocolate cake. This is a very important one. You use it all the time. If there's something wrong there, it's going to cause a problem. Whereas you might have another one for a salted Jell-O casserole that you're never going to use. And so if there's a problem there, you won't ever know because you were never intending to make that. But sometimes things are more than the sum of their parts. So maybe if all of your favorite dessert recipes were affected, now you can't make a dessert. So when we think about chromosome 15 in this region, it is a bit more than the sum of its parts, but there are some parts that are probably more important than others. And one that I want to focus on, as you know from the name of the talk, is UBE3A as a gene that's particularly important in this area. Again, it's one of the ones that's maternally expressed. We know that it's only turned on when it comes from the maternal copy. So that kind of fits with knowing that when there's an extra maternal copy, the symptoms are more severe. And we know that UBE3A is an important gene in the body. So it makes a protein that's called ubiquitin. And what happens is that you have some other protein that's doing its job in the body. Over time, it gets old, it gets degraded, and it's not working well anymore. So what ubiquitin does is it comes in, it tags this old protein and says that it should be recycled. So it's a long process of how this works, but it helps to regulate and make sure that everything is working well in the body by finding things that need to be removed. And we know that UBE3A is of particular importance because it's also a gene that leads to Angelman syndrome. In Dup15q, you have too much UBE3A, and Angelman syndrome, you have not enough UBE3A. But we need this delicate balance of having the right amount to keep our bodies working properly. And what's common to both Dup15q syndrome and Angelman syndrome is things like seizures and developmental delay, hypotonia, ataxia. So a lot of the biggest symptoms that we deal with are common to both, and this is the gene that they have both in common. So just in summary here, Dup15q syndrome is caused by having one or more additional copies of this 15q11.2 to q13.1 region, which is called the critical region. Most often it's a maternal extra copy that leads to the most severe symptoms. These extra copies can be within the regular chromosome 15, which is interstitial. It can be in an extra chromosome, which would be isodicentric or idic. And UBE3A is an important gene in this region that can contribute to the features of Dup15q syndrome. So again, this is a gene that for a couple different reasons seems to be of special importance, but taking that imprinting and all of those other things in mind, this is kind of jumping out as an important gene. So I'll kind of pause there and see if there's any questions in the time we have left.
Thank you. Oh, yeah, in the back, yeah. Thank you.
Oh, sorry. What specifically does the UBE3A and ubiquitin do for our bodies? Is that why we have digestive issues, that kind of thing?
Yeah, that's a good question. I think there's some other talks that are going to talk more about UBE3A that can get into more detail. I don't think that we can link UBE3A specifically of like it does this one particular thing in the body. It does affect a lot of our body. It affects our brains. So I think knowing some of these symptoms that are common among disorders that involve this is sort of clues of what it's doing. So again, affecting the brain in terms of seizures, development, and just being able to control like movement of our bodies is a big thing that it's doing. I think some other things like digestive issues and things like that sometimes are linked back to how well our muscles in our brain are controlling movements in the body. So like constipation, if your gut isn't moving properly and the muscles moving properly, can lead to constipation. So again, I think other talks will probably talk to it in better detail than I can.
Yeah. Hi, thank you for sharing. Can I just ask on the length of the duplication?
Yeah.
Is it that the longer the duplication, then has there been any studies that share that the length of the duplication affects the severity of the child, or it's not related?
And I think that the longer it is, it does impact severity. It kind of depends on exactly how long. There's some important genes in that region between breakpoint 3 and breakpoint 5, less so between breakpoint 1 and breakpoint 2, but there are some. I don't know if there's any specific studies that have looked at it. A lot of people on their lab reports don't get exactly where those breakpoints are, so it can be hard to get enough people to look at that. But I think in general what we see is like the larger it is, the more severe it tends to be. I will say, kind of on top of that, I think what impacts severity even more is how many copies of that main region you have. So having 4 copies is more severe than having 3 copies. So part of why we see in the isodicentric group that they tend to be more severe, they have usually 4 copies as opposed to 3 in the interstitial. Okay.
Hi, I'm a little confused because I think you said UBE3A was only expressed maternally, but isn't the level of UBE3A in neurons identical in maternal and paternal interstitial cases?
That's a great question. I don't know about that, but I can like reach back to you out later and check in on that, because we do know that there tends to be that imprinting. So yeah, but I don't know if there's an exception there. So. Okay, well, if that's everything, I think we can wrap up. I think if there's any other like specific questions about genetic reports, I am doing some like just one-on-one brief genetic counseling sessions coming up, and then a deeper dive into the subtypes of Dup15 in a breakout session later on. So thank you so much.